Hello, everyone, and welcome to today's webinar, High-throughput RNA-Seq of clinical samples using Covaris AFA ultrasonication. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Covaris. To learn more, visit covaris.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speakers, Dr. Stuart Levine, Director, MIT BioMicro Center, Massachusetts Institute of Technology, and Dr. Madan Ambavaram, Senior Application Scientist, Genomics at Covaris. For a complete biography on our speakers, please click on the Presenter tab. Dr. Levine, Dr. Ambavaram, you may now begin your presentation.
Thank you very much for the introduction. So I'm going to be talking today about our methods using the Covaris ultrasonicator for handling challenging clinically derived samples for Illumina sequencing, or short read sequencing. So I'm the director of the MIT BioMicro Center. We're an integrated genomics and bioinformatics core facility here at MIT. We work with labs across the campus, handling things everywhere from experimental design through library prep, sequencing, analysis, and even data management. Talk about how we are able to increase the power of NGS experiments using inexpensive replicates, and how we've been able to do this using low-volume library preparation. And then finally, how we've been able to use the Covaris sonicator to enable us to do high-throughput preparations of clinically derived samples. So this project really began with our experience as bioinformaticists with replicates, and the question of getting biological replicates done. So there are many studies out there that talk about biological replicates and how many replicates you can do. And when you talk to informaticists about this, the number they will give you vary widely. It can be the cast of a Broadway show, or maybe we're going to normalize across all of the people who've ever played Batman. On the other hand, when we ask how many replicates we see on a day-to-day basis, or saw on a day-to-day basis, it's not a number we wish to discuss, because it's certainly not a number we should be seeing. We want to see more replicates. And so the question was, how do we get there? That was the question that my lab undertook. And so in one of these papers, the Lee et al. From Bioinformatics, about a decade old now, they did a really nice experiment where they simply graphed some RNA-seq data. This is, in fact, from yeast. And they graphed the number of differentially expressed genes that they observed as compared to the number of reads per replicate that they saw. But what's interesting with this graph is that you can overlay it with a different set of data. So instead of varying reads per replicate, what if we lock in the number of reads per condition? And here we get these bands. And so I'm just going to walk briefly along this blue line here, which is 60 million reads. And so if we take those 60 million reads with a duplicate, we'd get 2 at 30, 3 replicates at 20 million reads, and 4 replicates at 15, and so on. And what we can see is that as we get higher numbers of replicates, even without increasing the number of reads that we're doing for the condition, we're getting significantly more statistical power. And so effectively, we now have 2 independent axes on which to increase our power. One, you can always do more sequencing, and you'll get more power that way. But also, just by increasing the replicate count, we can increase our power. And so how does that apply? So how do we then use this to make replicates routine? Well, the first, we can control those sequencing costs. Because we are locking the number of reads per condition, more replicates does not mean more money to Illumina or any of the other short read sequencing companies. Second, labor costs. Well, it doesn't actually cost us that much more to be able to do large batches than it does to do small batches. It's the same amount of time for the technician, even if we're doing a lot of the steps semi-manual. Third, failure costs. So if you have 3 replicates and 1 sample fails, now you've got duplicates. That's not publishable data. And so we have to go back and make sure that we're capturing every single sample. But because those samples, there are batch effects, we often will have to repeat the entire prep of all of the samples to capture even a few failures. Whereas if you have 5 replicates, 6 replicates come in, then the loss of a sample is no longer a big deal. We're able to just ignore the failed samples and continue on. So that leaves the final problem, which is reagent costs. And this is where we bring in miniaturization. So we do use some automation in this process. We use a classic automated liquid handler, the TCAM Freedom Evo. We have 2 of these. They are multiple decades old. Any liquid handler, fine, but these have 96 tip heads, liquid handling arms, and we use these for large volume pipetting. We also use an SPT LabTech Mosquito HV. This accurately pipettes between 0.4 to 5 microliters. We've been able to bring it down to about 0.2. We tend to do this in 3-4-well plates in order to minimize evaporation, but we're able to do low volume pipetting with this. So just as an example, we can begin with the classic Illumina library production, which is ligate immune PCR. Here we do all of the normal steps where you take randomized sheared DNA, you do end repair, A-tailing, then ligation of the Y-shape adapters, followed by amplification. And so this is one of the first methods that we focused on. And we did this using the Mosquito, where we would do on the blue methods that we were doing either by hand or on large scale, where we're quantifying and diluting the samples. Then we do an Ultra 2 library prep. This is using the NEB Ultra 2 kit at a 1:5 dilution. We then can clean this, quantify, normalize, and then we'll pool this also on the using automation. And so how does this work? Okay, so this works very robust. And so we can do libraries that can have inputs from 75 nanograms of input down to 20 picograms of input and get very reproducible traces. Now, the classic place we were focused on this was something like a chromatin IP, where you have very limited amounts of input. And so this is an example from that, where we're doing H2K4 trimethyl or a control whole cell extract with input amounts of 1 nanogram, 100 picograms, or 10 picograms. And you can see we're getting robust traces. We're getting similar numbers of peaks. And importantly, we're not only maintaining our library complexities, we're even getting higher library complexities. Now, what does this look like in terms of the peaks? This is 3 different example genes. This is from embryonic stem cells and so we have our classic active gene like a GAP-DH, where you have on the top in blue our old library construction methods that was with the Spryworks from Beckman Coulter, as well as the new miniaturized library preps in red, and with the higher amounts of input in the dark colors. And you can see that the traces on the red look very consistent as we go down, even down towards 10 picograms of input. UCP, that's with the highly active gene. UCP2 is uncoupling protein 2. This is a protein that is active in brown fat. It puts a hole in the mitochondria that allows the protons to escape immediately. That produces heat. It's not something you want in your embryonic stem cells, but it is a gene that is poised in these cells, so it's not actively transcribed, but the promoter is available and trimethylated. And here again, we can see robust signals, of course, in both samples at a nanogram, but even at 10 picograms of input, we're getting good signal with the NEB, more than we would with our classic method. The last gene to show you is the alcohol dehydrogenase cluster. In the ADH cluster is active mostly in our liver, but the ADH5 gene is constitutive, and we can see that here. So this is on in embryonic stem cells, and across the rest of this cluster, we expect to see silence. And that's what you want to see, is this line be very, very low. And we can see in the blue a bunch of these little spikes showing up, whereas in the red, we've got much more consistency and very, very few reads showing. Okay, so we have that, and sorry, the key thing out of this is the savings. So doing a singlet kit here would cost a lab about $100 per sample, but we're able to do this at about $1,500 for 96 samples. So we're getting about a 6-fold cost savings for all of the labs when we're doing this at batch. This is not a small impact. So if you were doing 16 samples, it costs you no more money to do 96. So we've expanded this method beyond classic LMPCR. We have a whole bunch of methods it works for. So both we can do classic LMPCR, we can do transposon-based, Nextera, XT, Nextera Flex. And we've been able, you can take this and we can do RNA on top of this as well. And whether it's we can do ribosomal depletion, poly-A pull downs, but also then fragmentation and cDNA generation, or SmartSeq-based template switching methods that will make full-length cDNAs, all of which we've been able to miniaturize. Okay, and this gives us a robust experiment. This is a control method one we did. This is again an RNA-Seq, where the miniaturized samples and the automated samples, they do separate from each other, but they're very, very similar, and a little different than the technicals, but they're very similar when you look at them overall. And that's kind of what you would expect. We don't expect methods to look exactly the same, but the biology is very easy to see. Okay, and we've used this for many, many different experiments. This is an example from the Griffith lab doing high throughput experiments with organs on chips, where we're able to isolate the RNA from a whole bunch of different areas, samples, and process them all in batch. Okay, so now I've talked about increasing our power through inexpensive replicates and how we've been able to use low volume library preparation to enable this. So now I'm going to move through how does we look at clinically derived samples and where does this come from? Well, in automation, we're expecting all the samples to be the same at the point we begin. And then samples come in when you're dealing with clinical samples, and this is what they look like. We need samples to come in that are all identical, and here we have samples coming in from clinically derived samples, and they're highly diverse. So what do we do in this case? Well, the challenge for RNA-Seq is that the very first step in all of these methods is the fragmentation of RNA using cations. And typically this is magnesium. And use heat in that and magnesium, and that results in the RNA fragment. However, this process will continue this hydrolyzation to hydrolyze the RNA down to mononucleotides. So you have to capture it in the middle. It's also sensitive to the buffer. Different amounts of EDTA in the sample will result in different amounts of free magnesium at different rates. So it's highly sensitive to the initial condition. When you're dealing with these precious clinical or clinically derived samples, having this type of variability is very stressful and concerning. So we asked whether sonication could work instead. So sonication works based on physics and on physical damage to the DNA based on length, that you're distorting the DNA and it's causing breaks in the DNA. But if the DNA and the RNA doesn't have a specific length, the efficiency of that process goes down. And so you can continue to add energy, you can continue that sonication process, and it will not continue to fragment anymore. So with this, we can take samples of both either high quality or lower quality RNA, put them in the machine at the same time, subject to the exact same amount of sonication, and you'll end up with fragmentation that's very similar. We use the Covaris R230. This is their newest machine. It uses AFA tube TPX plates. These are plastic plates, and we developed the methods with that. It processes columns at a time, allowing us to do whole plates very quickly. And because, again, it's non-destructive, it won't take it off to the point the sample is completely destroyed, we can put almost any input amount in to have this work. And it plugs directly into a broad spectrum of RNA-seq protocols, in theory, because that fragmentation is the first step of almost every one of these protocols. So does this actually work? We collaborated with Covaris on this. This was some of the very earliest pilots we did several years ago now, where we had different input amounts, whether they were from liver or from uterus with high and low amounts of input. And post-sonication, we ended with a single peak running around 200 base pairs. So we then did a pilot study. We took universal human reference RNA, and we degraded it with heat, different amounts. So we started with highly intact and brought it across a pretty broad range of quality. And then we either did the classic, which is you look at the protocol, you say how much different amount of time I should do with the magnesium. So we did that by hand, or we took all the samples and fragmented them on the R230. We then pooled all the samples back together and processed each one separately for Illumina sequencing. So here it's just to show you the data. This is the RNA as we fragmented it down across a whole bunch of different sizes using heat. And then this is what the libraries look like. And over on the left, we see all of the Covaris libraries are incredibly consistent, no matter what that starting material was. Whereas the by hand libraries are actually quite variable. And this is what we've seen classically. We then can take this and process it for two different methods. We were looking at one is the Ultra-2 NEP's ribosomal depletion method, as well as Takara's smarter stranded total RNA kit V2, pico input mammalian. We've always abbreviated this in our lab as ZAP-R, which is the key enzyme in that. And so the data I'm going to show you today is using 1 nanogram of input using the Takara ZAP-R kit. So this is the data that comes out. Here's just a raw correlation analysis. I will note the scale. This is a technical replicate. All of these should be completely identical. So our white on this scale is 0.9 correlation up to versus a 1. So these are all highly correlated. We're exaggerating that scale so you can see the difference. And you can see those samples in the manual block. Not even all of them worked well in library construction. Those that did are all well correlated, but not perfectly. Whereas the ones that are sonicated, we did it either treated the sample for a few minutes at 70 degrees, like you do on a bioanalyzer to denature it, or we didn't, we're getting highly consistent behaviors. We next look to see if there's any biases. Every type of RNA-seq we've seen to now is using magnesium. So is using sonication doing anything fundamentally different that we should be paying attention to? Doesn't mean it's wrong, just means it's different. So we started looking at things like transcript length, where we might expect the shearing forces to be different. And when we compare the log 2 TPM versus the length of the transcripts, there are obviously differences based on the transcript length, but whether we're doing a manual sonication in the red, or manual fragmentation in the red following the standard protocol, or the Covaris sonication in the blue, they're extremely similar across all of the different transcript lengths. How about UTR lengths? It doesn't matter in terms of UTR coverage at the 3' ends of genes, coverage. If they're short UTRs or long UTRs, we're seeing equal amounts of coverage, both on 5' UTRs and on 3' UTRs. GC bias, of course, there's always some GC bias with RNA-seq, but we're not seeing any difference with how we're doing the fragmentation. Maybe a little bit different at the high GC if we're doing the 70-degree treatment. How about classes of transcripts? Here again, we're really not seeing anything different, whether we're looking at protein coding genes, SNO RNAs, transcript process pseudogenes, and all of the other classes that are annotated. We're seeing very little difference. In fact, the biggest difference was whether we pre-treated it at 70 degrees or not before we put it in. Looking at an individual gene, this is an example, XBP1, it's on chromosome 22. And here's an example, these are sashimi plots that look at the splicing patterns that are observed. And if we look at the RIN3 sample versus the RIN8 sample, depending on the heat degradation, here we can see the splice pattern that is different between the detection, whether this particular exon is detected within the XBP1 gene. And when you do it by hand, we see it sometimes is and sometimes isn't. But with the Covaris treatment, we're seeing no difference depending on the starting concentrate, starting RIN. Okay, so that's a test case. What about real samples, real data? Here's that same data set. We processed this into libraries, and in fact, it's already been published. These were triple negative breast cancers, 62 samples that were shipped from Europe to the lab. The lab brought them in, and we were able to process these all in batch and had beautiful data, clustered beautifully, and was able to be published. Here's even more challenging. This was from 69 FFPE extracted RNAs that were from tumors of unknown origin. Here you can see the quality of the RNA is even lower. These we did the sonication on. And obviously when the fragments are even smaller than the sonication length, you don't rebuild them, but they do pile up very nicely. And then when we made the libraries, they all looked beautiful. This also has been recently published. And here again is the cluster data that shows how the data looks really robust for folks. So in conclusion, I've walked you through how we've been able to increase the power of our RNA-Seq experiments here through lowering the cost of replicates, and we do this by reducing the volume, and then how we've been able to use the Covaris sonicator to enable us to do the preparation of these clinically derived samples. And so I'd like to thank the team who actually did the work, folks on my team, Tom Volkert over at the Whitehead Core, who provided us the methylation samples, the labs who were willing to be our guinea pigs as we did the technical development, as well as the folks at SPT and Covaris who helped us with developing this method. And thank you for your attention. And I'll now pass it over to Madan for his talk.
Hello, everyone. Thank you, Kelly, for the kind introduction. Also, thank you, Stuart, for the wonderful and enlightened presentation. My name is Madan Ambavaram, and I am the Senior Application Scientist at Covaris. It gives me immense pleasure to present today alongside Dr. Stuart Levine. I would like to take this opportunity to discuss about RNA sharing solution using Covaris proprietary technology, adaptive focused acoustics, AFA in short. Let me start by sharing the Covaris vision. At Covaris, our global solution mission is clear: to help more people worldwide by delivering advanced sample prep solution that enhance our understanding of biomolecules, ultimately improving and saving lives. Covaris adaptive focused acoustic technology offers high-value solutions in the pre-analytical sample prep domain. Our technology enables highly efficient extraction and processing of biomolecules from a whole range of different sample types. This processing is done in a very robust and reproducible fashion that enables streamlined workflows across different omics pipelines. It all starts with the use of high-frequency ultrasonication AFA method for delivery of acoustic energy in a very focused manner. This is a non-contact process, thus eliminating any chance of contamination. The AFA process is extremely tunable and thus offering significant operational flexibility. The entire process is conducted in a thermally controlled environment, thereby ensuring integrity of the biomolecules. The different configurations and the superior result from the AFA process allows the user to fit all the puzzle pieces together by offering a single technology platform that allows for highly quality performance, low cost per samples, as well as the versatility needed to accommodate all current and future needs. The high quality data output from the AFA process helps provide deeper insights, be it in oncology, in tumor transcriptomics, biomarker detection, fusion detection, and pathway analysis. All these insights lead to actionable data that helps in improving and saving lives. That is the objective of Covaris. Speaking about the challenges in RNA fragmentation or RNA sequencing workflows in general, one of the major challenges is biased fragmentation. Achieving a consistent and specific size distribution of fragmented RNA is pivotal for successful library prep, which needs significant investment of time and resources to standardize the length of RNAs. AFA provides tunable fragmentation, allows the user to generate fragment sizes across a wide range while ensuring greater repeatability and reproducibility. Sample-to-sample variability is also one of the major concerns in the clinical and environmental space. AFA is agnostic to the sample type. And speaking about workflow compatibility, there are multiple protocols depending on sample origin, quality, and input amounts. AFA is compatible with low input samples and various ribosomal RNA depletion workflows, which is important to maximize the sequencing efficiency. And also another challenge is workflow implementation. Considering variability in sample type, non-AFA methods need longer optimization time, particularly for scalability and adaptability to new kits or sequencing methods. Using Covaris AFA, different sample types can be loaded on the same plate and processed with the same AFA method protocol, which allows streamlined protocol with minimal or no optimizations. These benefits will ultimately translate to lower sequencing cost on the back end. At Covaris, we provide the best RNA fragmentation solutions, which improves economics, performance, and the workflow ease of use. As you can see here, these three pillars are the foundations of our focus and dedication to the genomics community. We look at the ease of use and adoption of our solution, performance improvement, and better economics. The claims presented here are not just words. They are backed by data and real case studies from our customers and collaborators. One such application using AFA for tumor transcriptomics, earlier in this webinar, you have heard from Dr. Stuart S. Continuing with the reproducible RNA sharing using different sample type, here is an example demonstrating AFA-based RNA sharing, which is agnostic to the sample type. For example, RNA from cell culture or FFPE tissue or synthetic mRNA or double-stranded RNA. AFA-based RNA sharing produces highest yield and quality of library for sequencing compared to other processes such as cation-based fragmentation. A representative RNA electropherogram prior to AFA treatment is shown on the top. The bottom electropherograms are after AFA treatment. Depends on the library specifications and workflow requirement, the fragment sizes are tunable with a high reproducibility. Speaking about the scalability aspect, Covaris instruments are enabled to process from 1 sample to 384 samples based on workflow and throughput requirements. Here is one of the case studies from our customer and collaborator. Greenlight Biosciences have used AFA-based fragmentation for the mRNA and double-stranded RNA library prep workflows. The key takeaways from this study is that they have obtained successful and consistent reproducible fragmentation using single fragmentation protocol, which is pivotal for successful QC and library prep of high-throughput workflows, and also have obtained excellent sequencing data, which is more than 99.8% mapped reads from both mRNA and double-stranded RNA libraries, with excellent run performance and less sequencing errors compared to cation-based libraries. Also, they have observed better coverage at quantic level, helps to evaluate the gaps in genome assemblies. Covaris offers a streamlined RNA sharing workflow with standardized protocol, especially when dealing with a low input and wide range of sample concentrations. In addition, AFA solutions offer different consumable formats to address different sample processing throughput requirement. For example, low throughput 1 to 8 samples, or high throughput 96 or 384 well, or batch processing. In summary, Covaris offers a robust, reliable, and reproducible workflow for RNA sharing, exhibiting a single fragmentation workflow for various sample types with minimal or no optimizations, suitable for high-throughput RNA library prep and workflow miniaturization with significant cost savings. And also, AFA is highly compatible with ribosomal RNA depletion workflows, meaning to say all in one solution by plugging into a broad spectrum of RNA sequencing protocols. In a nutshell, why Covaris? Covaris has been the gold standard for DNA shearing. Market recognition of our outstanding sample prep and DNA RNA fragmentation capabilities is evident in several NGS library prep workflows, but in clinical, pharma, and R&D sector. Our solutions reduce hands-on time and the financial burden of the lab. Also, Covaris instruments coupled with consumables and reagents are easily integrated in many NGS workflows, thereby easily meeting current and future high-throughput needs. We are working towards holistic sample prep solution here to solve all your sample prep challenges. Please contact us to discuss your next project or how you want to improve your current lab's data. Once again, thank you all for your attention.
All right. Well, thank you so much, Dr. Levine and Dr. Ambavaram, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. We've already got some great questions coming in here. And this first question is for Dr. Ambavaram. This question asks, what have you seen in terms of uptake, and how many people have moved over to Covaris-based RNA shearing method?
Thank you, Kelly. First of all, it's a very good question. In terms of uptake, yes, using Covaris for RNA sharing workflows is in positive trajectory. About how many people are using, I would say there is an increasing demand year over year using AFA-based RNA sequencing workflows in both pharma and biotech sectors. Thank you.
Great, thank you. All right, this next question here is for Dr. Levine. How many advancements in RNA-Seq contribute to more effective patient stratification and treatment in the coming years?
Thank you for the question. It's a challenging question because we have had a long experience with RNA-Seq on a single patient level. And a lot of the challenges are isolating the proper samples to work with. You can do things like PBMCs and the like, or samples from tumors. Part of the challenge has been the speed at which you can get the data. Part of the challenge has been interpreting that data in a way that's meaningful to the researchers. Being able to standardize and lower the price of that type of RNA-Seq, I think would be helpful in helping use this data more actively in the clinic. There are places it's probably much more relevant. So cancer, for example, is an area which is a disease of the genome. Frequently we're looking at mutations, but some of the challenges associated with cancer is that it can be changes in the epigenetics as well, which can result in changes in RNA or changes in splicing, but not as obvious in the genetic changes. And so being able to look at the RNA can possibly provide a more accurate diagnosis, or if not diagnosis, then suggest which therapeutics may be more effective.
Great, thank you. All right, another question for you, Dr. Levine. This question asks, how has the development of RNA-Seq technologies, specifically in whole transcriptome sequencing, contributed to our understanding of complex gene expression patterns? And what are the major challenges that researchers still face in accurately profiling entire transcriptomes?
Thank you for the question. The whole transcriptome sequencing, or whole transcriptome is RNA-Seq, or is really just the continuation of microarray technologies that we've been doing for for several for several decades now, and is really fundamental, but for our understanding of what of systems biology, what defines a cell, what defines a cell type. So it's absolutely fundamental to a huge amount of our current understanding of cell biology. So, what are the challenges that remain? There are several. First is that as a question of what RNA-seq tells you is the steady state level of RNA in the cell. That's a useful piece of information, but it's kind of a capture of some information that's you often want. What's the rate of transcription, which is more correlated with the genome? What's the rate of destruction? What's the rate of translation? You can also look at questions like things that need to be done. Looking at small RNAs is an area where the current technologies are very limited. We've done some work looking at that, and the correlation of we do not get an absolute measurement of small RNAs. And so we see very little of it actively in our lab, mostly because it costs about the same as total RNA-seq, and you're not getting anywhere near as much information. So I think there's a lot of questions still. Areas like single cell also are really important because those allow us to detangle the different cells within a population. Bulk RNA-seq tends to be very homogenized. You can't tell. Sometimes signals are lost based on where you're looking at, if the signal is coming from changes in expression in a single type of cell within a population instead of the whole population. But what it does give us is a very inexpensive way to look at RNAs and cells, particularly with the high throughput methods. We can do almost 96 samples for the cost of a single sample for RNA-seq, for single cell.
Wonderful. Thank you, Dr. Levine. I have another question for you here. How critical is a normalized RNA input amount in your miniaturized reactions?
Being perfect on that is not critical. We do the normalization afterwards. So Covaris in particular is very helpful for this. One of the real challenges with the reasons you need that normalization is to have the right amount of time with the magnesium so that you get consistent fragmentation. If you have too little RNA, it will be completely destroyed. The Covaris allows that to be, because it's relatively insensitive to the amount of input, or quite insensitive to the amount of input. And so you can use the same protocol and get very different amounts. Now, you don't want something 10-fold or 100-fold different because all the rest of your reagents are going to be way out of whack. But it's less important that you're perfect in that reaction. So yeah, it was a good question.
Great, thank you. All right, we have time for another question here, so I'll ask another one. This one asks, question for both, showing Covaris shearing is superior to cationic or heat fragmentation. Dr. Ambavaram, can you answer this one?
Mm-hmm. Yeah, sure. Yeah, it's a good question. Based on the in-house data and the customers who have been seeing, AFA-based RNA fragmentation is very evident and clearly showing that the data is very superior, which is evident by many technical publications and peer-reviewed journals. Thank you.
So for myself, I would say if you're doing one sample, you can get it to work, right? We've been doing that for ages. It's not super hard. But if you look at the protocols, they all begin by saying, optimize your fragmentation. Okay? And that doing that optimization takes time. It is also sensitive to the buffers, like I said, with the EDTA and other things coming in. And so it's hard to predict exactly what it will be, but it's going to work, provided you have enough sample. The Covaris allows us to kind of just stick our fingers in our ears and say, la, la, la. As long as the sample buffers are, we do a cleanup of the RNA beforehand to make sure there's nothing weird in the samples. We've gotten very consistent results. And so we don't have to worry as much about it. Beyond that, I was hoping to show that the quality of the RNA-seq data is very similar to the quality that you get using cationic fragmentation. Is it better? Is it worse? It's a very hard thing to say because it's very hard to end up with a gold standard for any of the bulk RNA-seq methods. Any of the different RNA-seq methods are going to have different noise parameters, and that results in clustering quite separately. So it's kind of hard to know exactly what the gold is. The same is going to be true for qPCR. That's not a true gold standard once you're trying to get an absolute value.
Fantastic. Well, thank you both for that answer. That will conclude our session for today. Unfortunately, we are out of time. So I want to ask again, Dr. Levine and Dr. Ambavaram, do you have any final comments for our audience?
Well, thank you all for joining the webinar, and it's a great opportunity to present our Covaris technology. Thank you.
Thank you again. Thank you to the collaborators who've supported us, as well as to Covaris for all of their help in developing the protocol and the opportunity to speak today.
Well, thank you again, Dr. Levine and Dr. Ambavaram, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Covaris, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.